Welcome again to Cancer as a Mitochondrial Metabolic Disease Basics. Most people have heard that cancer cells utilize glucose or are dependent on glucose, but fewer people realize that tumors are equally or even more dependent on another fuel source, glutamine. So, does glutamine feed cancer? In this video, we're going to break down what glutamine actually does inside cancer cells, how it supports their growth, and what the evidence actually shows. So without further ado, let's get into it. Okay, does glutamine feed cancer? What the evidence says. So I'm going to start off by going over a couple of papers that came out in 2012 and 2017, respectively. One was titled Metabolic Reprogramming in Cancer, Unraveling the Role of Glutamine in Tumorigenesis. And it says increased glutaminolysis, or the breakdown or utilization of glutamine, is now recognized as a key feature of the metabolic profile of cancer cells, along with increased aerobic glycolysis, the Warburg effect. In this review, we discuss the role of glutamine in contributing to the core metabolism of proliferating cells by supporting energy production and biosynthesis. And in the next paper, glutaminolysis, a hallmark of cancer metabolism, it says glutamine is the most abundant circulating amino acid in blood and muscle and is critical for many fundamental cell functions in cancer cells, including synthesis of metabolites that maintain mitochondrial metabolism, generation of antioxidants to remove reactive oxygen species, synthesis of non-essential amino acids, NEAAs, purines, pyrimidines, which make up DNA and RNA, and fatty acids for cellular replication and activation of cell signaling. And it specifically says in the paper later on that despite remarkable heterogeneity in cancer metabolism, certain characteristics, such as increased glucose, which we talked about in the last video, and glutamine consumption, are broadly conserved, a phenomenon that indicates fundamental biochemical necessities and that is exploited for clinical imaging in tumors with PET scans, like we said in the last video, with labeled metabolite analogs. The next paper we're going to go over is Rethinking Glutamine Metabolism and the Regulation of Glutamine Addiction by Oncogenes in Cancer. And it was published in March of 2023. And it says glutamine, the most abundant non-essential amino acid in human blood, is crucial for cancer cell growth and cancer progression. Glutamine mainly functions as a carbon and nitrogen source for biosynthesis, energy metabolism, and redox homeostasis maintenance in cancer cells. Dysregulated glutamine metabolism is a notable metabolic characteristic of cancer cells. Some carcinogen-driven cancers exhibited a marked dependence on glutamine, also known as glutamine addiction, which has rendered the glutamine metabolic pathway a breakpoint in cancer therapeutics. However, some cancer cells can adapt to glutamine unavailability by reprogramming metabolism, thus limiting the success of this therapeutic approach. More on that later. Given the complexity of metabolic networks and the limited impact of inhibiting glutamine metabolism alone, the combination of glutamine metabolism inhibition and other therapeutic methods may outperform corresponding monotherapies in treatment of cancers. This is important because this is exactly what ketogenic metabolic therapy proposes to leverage, is not just a monotherapy, but a combination of therapies in a systematic fashion. We'll get into that later. This review summarizes the uptake, transport, and metabolic characteristics of glutamine, as well as the regulation of glutamine dependence by some important oncogenes in various cancers to emphasize the therapeutic potential of targeting glutamine metabolism. Furthermore, we discuss that glutamine metabolic pathway and glutaminase 2 pathway has been substantially overlooked. Finally, we discuss the applicability of polytherapeutic strategies targeting glutamine metabolism to provide a new perspective on cancer therapeutics. And it says here in the introduction of the paper, metabolic reprogramming is a hallmark of cancer that manifests in several ways. Cancer cells exhibit substantially enhanced glucose uptake and full utilization of glycolysis, TCA cycle to generate large amounts of ATP, Warburg effect. We talked about in the last video in the question, does cancer require and hyperutilize glucose? That we did go over. Additionally, cancer cells may exhibit accelerated uptake, transport, and metabolism of glutamine. which is the most abundant non-essential amino acid in the human body. Adaptation to rapid metabolism is achieved by regulating genes that encode metabolic drivers to increase expression of favorable transporters and metabolic enzymes. Furthermore, metabolism regulating interactions occur between cancer cells and the tumor microenvironment, TME. Since Hans Krebs first studied glutamine metabolism in animals, the biological role of glutamine in cellular growth and cancer cell biology has gradually been recognized. In 1955, Eagle discovered a high dependence of cancer cells on glutamine, also known as glutamine addiction. Specifically, and this is where it gets pretty important, specifically, cancer cells consume 10 to 100 fold more glutamine than they do other amino acids. Glutamine is the most abundant non-essential amino acid in the bloodstream. High levels of glutamine can promote cancer cell proliferation by serving as nitrogen and carbon sources for biosynthesis of nucleotides, fatty acids, and non-essential amino acids. In addition to its primary role in macromolecular biosynthesis, glutamine is also involved in the cellular uptake of essential amino acids and the maintenance of the mitochondrial membrane potential and the production of glutathione and nicotinamide adenine dinucleotide phosphate, which we are talking about heavily in the main playlist when we're talking about the maintenance of redox homeostasis. If you want to go over there to that playlist, go ahead, which are required to maintain redox homeostasis. Altered glutamine metabolism is a significant outcome of changes in energy metabolism in cancer cells. So, I think that we have established in these first couple of slides that glutamine is also heavily dependent upon by cancer cells, on the order of 10 to 100 fold more compared to other amino acids found in the bloodstream. The next paper, which was fairly recently published in April of 2025, called The Warburg Hypothesis and the Emergence of the Mitochondrial Metabolic Theory of Cancer, states in the abstract, Otto Warburg originally proposed that cancer arose from a two-step process. The first step involved a chronic insufficiency of mitochondrial oxidative phosphorylation, OXPHOS, while the second step involved a protracted compensatory energy synthesis through lactic acid fermentation. His extensive findings showed that oxygen consumption was lower while lactate production was higher in cancerous tissues than in non-cancerous tissues. Warburg considered both oxygen consumption and extracellular lactate as an accurate marker for ATP production through OXPHOS and glycolysis, respectively. Warburg's hypothesis was challenged from findings showing that oxygen consumption remained high in some cancer cells despite the elevated production of lactate, suggesting that OXPHOS was largely unimpaired. New information indicate that neither oxygen consumption nor lactate production are accurate surrogates for quantification of ATP production in cancer cells. Warburg also did not know that a significant amount of ATP could come from glutamine-driven mitochondrial substrate-level phosphorylation in the glutaminolysis pathway, with succinate produced as an end byproduct, thus confounding the linkage between oxygen consumption and the origin of ATP production within mitochondria. Now, I get that for this basics playlist, that was a lot of jargon and a lot of probably concepts that you never heard of before. The most important piece, I would say, of this particular abstract, which we're going to hyper focus on for this video, is going to be right here, where it says that Warburg also did not know that a significant amount of ATP or energy, cellular energy, could come from glutamine-driven mitochondrial substrate-level phosphorylation in the glutaminolysis pathway with succinate produced as the end product, thus confounding the linkage of oxygen consumption and the origin of ATP production within mitochondria. We're going to unpack that because within this paper, which was an awesome paper, by the way, Seyfried and his group come up with these questionable assumptions. And in questionable assumption number 3, it says that OXPHOS and lactic acid fermentation are the only sources of ATP production or energy production within cancer cells. Basically, what they're saying is it's either going to be oxidative phosphorylation through energy metabolism through mitochondria, which is normal metabolism, or it's going to come from lactic acid fermentation, glucose being converted to lactate. And that's saying, and what they're saying is this is a questionable assumption, and they're going to try to tear that down. And it says here, neither Warburg nor Winehouse knew that significant ATP or energy could be produced through glutamine-driven fermentation or mitochondrial substrate level phosphorylation in the glutaminolysis or the breakdown of glutamine pathway with succinate generated as the end product. Just like in glucose fermentation, when lactate or lactic acid is the end product of that pathway, succinate is the end product of the glutamine fermentation pathway, thus confounding the linkage between oxygen consumption and the origin of ATP production within tumor cell mitochondria. Wow, glutamine has been largely recognized as an anaplerotic, which means that this glutamine, as in these other papers are saying, it's important for nucleotide synthesis, for protein and amino acid synthesis, for fatty acid synthesis. Basically, that glutamine is just being used as a raw material. What they're saying is, what Seafried and others are saying, is that it's not just this anaplerotic source of raw materials, but it's actually being fermented just like glucose is, but inside the mitochondrial TCA cycle. New findings show that glutamine can also be fermented for ATP production via mitochondrial substrate level phosphorylation. And I'm going to kind of hammer this home, or this point home, for is cancer fed by glutamine also? I'm going to use this paper that was published in 2024. And it says amino acid and a glucose fermentation maintain ATP energy content in mouse and human malignant glioma cells, basically brain cancer. And it says the result showed that glutamine was a source of ATP content respective of oxygen. This is important because most of the tumors don't have a lot of oxygen. No other amino acid could replace glutamine in sustaining ATP content and viability. ATP content persisted in the absence of glucose and under hypoxia or low oxygen in conditions, which is more true to where cancer actually lives, ruling out substantial contributions through either glycolysis or the Warburg metabolism or oxidative phosphorylation, OXPHOS, under these conditions. Mitochondrial complex 4 inhibition showed that oxygen consumption does not accurately measure for ATP production through OXPHOS. And then later in the paper, it says the addition of glutamine, 55%, glutamate 42%, and glycine 7% increased bioluminescence. They're basically seeing what amino acids is actually keeping these cancer cells alive relative to the baseline after 24 hours. And at 48 hours, glutamine was the only amino acid that actually kept these cancer cells alive from an energetic perspective. So this paper also had a great graphic associated with it. And basically, I'm going to distill down what we have seen in the last two basic videos. We see that glucose is heavily dependent upon by cancer cells. We saw that in the last video, 10 to 30x more than baseline or normal cells is how much glucose is being used. And that glucose, at least in part, is used to make energy and is fermented into lactic acid. ATP is the energy portion of it. It does other things too, which we'll talk about kind of one at a time so we don't get too lost in the weeds here. But glucose makes energy, ATP, through the fermentation into lactic acid. What we're also basically showing is that in addition to glucose, glutamine, a non-essential amino acid, is also highly responsible and dependent upon by cancer to make energy as well as other things. And the end product of that metabolism is the fermentation of glutamine into succinic acid, which is a different type of acid, both of which will be transported outside the cell to aid in the creation of the acidic tumor microenvironment, which has its own issues that we'll have to unpack. But my point is that cancer relies on two fuel sources: glucose, we talked about in the last video, and glutamine. And we saw in this video that glutamine is being uptaked and utilized 10 to 100-fold more than any other amino acid, which is substantial. And it's at least partially true because glutamine is the highest concentration of any amino acid in the bloodstream. So it's readily available compared to other amino acids, which are much less available in the bloodstream. I hope this answered the question, does glutamine feed cancer? Yes, it feeds cancer. And just like glucose, it doesn't just feed cancer, it helps it grow in many different ways. But at the end of the day, cancer is just as reliant on glucose as it is glutamine. And in some ways, it may be even more reliant on glutamine. and only time will tell how much that is and how that varies depending on tumor to tumor. If you like videos like this, please like, share, and subscribe. Until next time.